Welcome to Beyond Biology Lab YouTube channel. Today, we explore a fascinating molecular anomaly, how common anti-inflammatory drugs can somehow force immortal leukemia cells to reawaken their dormant suicide programs. In oncology, overcoming the cellular resilience of malignancies is a profound challenge. Relying purely on the development of entirely new synthetic agents is limited not just by long timelines and clinical hurdles, but by massive economic challenges that currently stifle developers and laboratory startups. But what if the blueprint to selectively bypass this immortality was hiding in the mechanisms of everyday drugs?
Let's examine this explainer of research by Singh and colleagues, which uncovers a hidden vulnerability in malignant hematopiosis. Published in the journal Apoptosis, this paper investigates how two common non-steroidal anti-inflammatory drugs, specifically Sulindac sulfide and Diclofenac, exert a profound anti-proliferative effect on acute myeloid leukemia, or AML cells. This is a grounded, preclinical investigation into the precise signaling pathways that dictate cellular life and death.
Today, we'll be covering the AML-Apoptosis paradox, how NSAIDs trigger leukemic death, the genomic and proteomic clues, the AP1-JNK pathway, validating the molecular targets, and finally, future combinatorial therapeutics.
Section 1. The AML-Apoptosis paradox. The crucial point here is that AML cells survive by actively disabling their intrinsic safety mechanisms. Now, acute myeloid leukemia is a remarkably heterogeneous disease. It's driven by a wide array of genetic alterations. But the common thread across all these distinct types of leukemia is apoptosis evasion. The leukemia-initiating clone basically gains a proliferative advantage over normal hematopoietic stem cells because, despite massive genomic instability, it refuses to trigger programmed cell death. So, the central question Singh and his team posed was this: what if those dormant death pathways are actually still structurally intact and could be selectively reawakened through a common pathway?
Section 2. NSAIDs trigger leukemic death.
The data here shows remarkable consistency across 42 de novo AML patient samples, as well as established cell lines like THP1 and HL60. When the researchers exposed these cells to 100 micromolar concentrations of Sulindac sulfide and Diclofenac over a 48-hour period, they observed dramatic spikes in apoptosis fold change. For example, in THP1 cells, Diclofenac induced a 6.30-fold increase in apoptosis, alongside a corresponding drop in viable cell numbers. This provides robust evidence that these specific NSAIDs can actually force these highly resistant cells to initiate their own destruction.
Section 3. Genomic and proteomic clues.
Through differential 2D gel analysis of HL60 cell lysates treated with Diclofenac, the team found that 211 protein spots significantly changed their expression levels. Using MALDI-TOF mass spectrometry, they identified components linked to cytoskeletal reorganization and cellular metabolism. But critically, they identified a marked increase in the caspase-3 precursor. Since caspase-3 is a primary executioner enzyme in programmed cell death, this provided the first major clue that the MAPK/JNK signaling pathway was being activated.
Complementing the proteomics, gene expression microarrays and quantitative RT-PCR pinpointed a massive transcriptional activation of the AP1 transcription factor family members, specifically c-Jun, Jun-B, and Fra2. Furthermore, they observed a robust induction of the growth arrest and DNA damage-inducible gene GAD45-alpha. In some patient samples, c-Jun expression was actually induced nearly 100-fold by Sulindac sulfide. This genomic data beautifully linked the initial drug exposure to the downstream executioner proteins.
Section 4. The AP1-JNK pathway.
This maps the sequence of the cellular execution. It starts with NSAID exposure. This external trigger induces the expression of AP1 transcription factors, which then combine to form active heterodimers. Next, these heterodimers directly drive the transcription of GAD45-alpha. Once activated, GAD45-alpha serves as a critical upstream signal that activates the c-Jun N-terminal kinase, or JNK. Finally, JNK activation leads directly into caspase-3-dependent apoptosis. It's a highly ordered cellular pathway that entirely bypasses the cancer cell's normal evasion tactics.
Section 5. Validating the molecular target.
To confirm that JNK was truly the linchpin of this entire process, researchers introduced a specific JNK inhibitor, SP6001, to the NSAID-treated cells. By applying this inhibitor,
The pro-apoptotic potency of the NSAIDs drops significantly. In THP1 cells, apoptosis decreased by up to 51%. In primary patient samples, it dropped by an average of 26 to 40%. This proves that JNK activation plays an integral, indispensable role in executing this targeted cell death. Furthermore, the upstream components were tested using molecular genetics. Transient overexpression of GAD45-alpha or specific AP1 heterodimers, like c-Jun paired with Fra2, was sufficient to trigger apoptosis all on its own, without any drugs present at all. Conversely, when RNA interference was used to knock down Jun-B and Fra2, it effectively rescued the leukemic cells from NSAID-induced death. This bidirectional validation neatly isolates the exact molecular circuitry responsible for the phenomenon.
Section 6. Future combinatorial therapeutics.
By exposing how NSAIDs induce AP1 and GAD45-alpha to trigger JNK-mediated death, researchers have illuminated a highly conserved pathway that exists across genetically distinct subtypes of AML, including those with complex karyotypes or FLT3-ITD mutations. It proves that the cellular machinery for apoptosis isn't missing in these cancers. It's simply dormant, and it can be pharmacologically reactivated.
As the authors note, this isn't about framing NSAIDs as a standalone cure for leukemia. Rather, it presents a scientifically grounded blueprint for next-generation combinatorial medicine. By utilizing agents that activate this AP1/GAD45-alpha axis, we can amplify the efficacy of conventional cytotoxic drugs. For laboratory startups and developers facing immense economic challenges in discovering novel oncology drugs, finding ways to intelligently repurpose established compounds to break cellular resistance offers a highly viable, cost-effective path forward for biological therapeutics.
Identifying a single, dormant pathway fundamentally changes our perspective on cellular resistance, offering a sophisticated framework for targeted medical interventions. Thanks for watching. Please like and subscribe Beyond Biology Lab YouTube channel.